This is podcast number 419, and we'll be talking about photosynthesis. You've talked about this in other classes, maybe not to the extent of this class, even though most of the emphasis is on glycolysis and catalysis, breakdown of products to make energy. This is arguably the more important pathway to be committed to memory, because this is the pathway by which arguably most of the life on the planet depends. There's a few microbes that depend on chemosynthesis in deep sea thermal vents, and then other organisms that depend on those organisms. But by and large, most of what you think of as alive depends on this process. And the anatomy of the chloroplast is unusual. As we've said before, there's 3 membranes. So 2 outer membranes and 1 inner membrane. I've actually had to correct this as an editor on some advanced cell phys textbooks because they had it wrong. But mitochondria have 2, chloroplasts have 3. Okay, the outer mitochondrial membranes have integral membrane proteins that allow macromolecules to pass through them. They're called porins. They're very much like mitochondria. And if something is less than 5,000 molecular weight, it can pass through those porins. Bigger than that, you need a more specific transporter. The light reactions are going to happen on the innermost membrane, which is called the thylakoid membrane. They're called the light reactions because they depend upon light, although it's confusing because as soon as the light goes off, these reactions stop. And then the Calvin cycle, which uses the products of the light reactions, the light-dependent reaction, happens out here in the stroma. So you have the 2 outer membranes, and then you have the innermost membrane. That innermost membrane is called the thylakoid membrane. A stack of thylakoid membranes like this is called granum. Several stacks are called grana. They look like pancakes, and your grandma makes the best pancakes. So hopefully that'll help you remember. So several stacks are called grana. The individual membrane is called the thylakoid membrane, and that's where the light-dependent reactions happen. The space outside the thylakoid membrane is called the stroma, and then the space inside the thylakoid membrane is called the lumen. And you're going to see a lot of similarities to how electron transport happens in mitochondria. Okay, so these are false color transmission electron micrographs of chloroplasts, and you can see stacks of thylakoid membrane here, grana, and then individual thylakoid membrane here. It's really hard to see the 2 outer membranes because they're so close together, but there are 2 outer membranes, and then this is the 3rd membrane on the inside, and the space outside the 3rd membrane, but inside the 2 outer membranes is called the stroma. Okay, so real life and then a stylized diagram. Again, if I asked you to draw a chloroplast, don't try to draw something 3D like this. Just draw 1 line per membrane. So you'd have 2 outer membranes and then 1 inner membrane all folded up as a thylakoid membrane. All right. Integral to the function of the chloroplast is a light-absorbing pigment called chlorophyll. Chlorophyll has a very similar structure to hemoglobin in that it has a heme group, but instead of iron at the middle of that ring, it's magnesium. And this ring contributes to how chlorophyll will absorb light. And when it absorbs light, the electrons in this molecule enter a higher energy orbital. Okay, these are energy levels where electrons are most likely to be found. You have s orbitals, p orbitals, and so on, different shape orbitals. When photons get absorbed by the chlorophyll molecule, their electrons enter a higher energy orbital, and then they'll fall back down to where they started. And when they fall back down to where they started, their original orbital, they'll release the energy that they absorbed. Okay, they can release that energy as heat, or they can transfer that energy to another pigment that is nearby, and ultimately transfer that energy to a reaction center chlorophyll, which will actually, instead of the electrons just moving from one orbital to the next, they'll actually leave the atom. And that is key to life on Earth. So we talk about photosystems in photosynthesis, and photosystem is a protein chlorophyll complex that is embedded in the membrane that contains multiple different proteins and maybe multiple different pigments, such that there are chlorophyll molecules that are absorbing light, and these are called antenna pigments. So any given photon hitting a leaf, hitting a photosynthetic organism, is not necessarily going to excite all the chlorophyll molecules at once. Some get excited, some don't. So these are arranged like an array, and they are very much like an antenna, and that something out there is going to pick up that photon. Then if those electrons get excited and they go to the higher energy level, when they return to their original energy level, they can give off that absorbed energy as heat, like we just saw in the previous slide, or they can transfer that excitement, that energy, into another antenna pigment, which then goes through the same process. The second one, their electrons get excited, go to a higher energy level, back down to the original energy level, and that bounces back and forth until eventually they're going to be in proximity of what's called the reaction center chlorophyll. And when the reaction center chlorophyll absorbs that energy through resonance transfer, instead of the electrons coming back to their original energy level, the electrons actually leave this chlorophyll molecule and get transferred onto an electron acceptor in an electron transfer chain. An electron acceptor being similar to the molecules that we talked about already, like NAD or FAD, something that it can accept high energy electrons and then pass them off to something else. So once again, the plant absorbs light. Those electrons get excited in their chlorophyll molecules. They cause the electrons to go to a higher energy level. They return to the original energy level, and the energy that is absorbed is given off. It can be given off as heat, or it can be transferred to another pigment. When it's transferred to other pigments in the array, eventually that will transfer to the reaction center chlorophyll, which now is not going to transfer it to another pigment, but instead the electron is actually going to physically leave the molecule instead of just going to a higher energy level and coming back. It's going to leave the molecule and go to an electron acceptor. If you look at the ability of different chlorophyll pigments and non-chlorophyll pigments to absorb light, and you put extracted chlorophyll in a spectrophotometer and you tune the spectrophotometer to 400 nanometers, and you say, okay, how much light can you absorb at 400 nanometers? In the case of chlorophyll B, that's not much. and then you go to 410 and 420 and 450, 460, 500. So around 480 nanometers, the amount of light that chlorophyll B can absorb is the most, and then it decreases at higher wavelengths, but there's another peak around 640. And then chlorophyll A has a different absorption spectrum in that it absorbs maximally at different wavelengths. And then if you look at the ability of the plant to do photosynthesis, you can do the same sort of thing, and you shine 400 nanometer light on the plant and you measure how much oxygen is producing. Oxygen is a byproduct of photosynthesis, and you keep doing that for each wavelength and you can get this sort of a graph. And you can see that the absorption spectrum here is very similar to the action spectrum down here, in that the plant is absorbing light of certain wavelengths, and those wavelengths are the things that are driving photosynthesis. There are not just chlorophyll pigments in most higher plants, but there are lots of other kinds of pigments in higher plants also. They overlap in their absorption spectrum, and they all contribute to the action spectrum shown. Well, actually, no, not shown here. But they do overlap with the action spectrum shown here for photosynthesis. This black line here is the sum of all the light that is reaching the surface of the planet, and the photopigments that plants are expressing in order to capture that light overlap so that they are capturing most of the big peak of light that is coming from the sun. But notice down here, they're not all chlorophylls. So we tend to talk a lot about chlorophyll. Chlorophyll is important, but there are other pigments also. And those other pigments start to be seen about this time of year. So leaves in the fall are changing color. A lot of that is due to the dominance of chlorophyll no longer being so dominant. So these other pigments, like phycocyanins and phycoerythrins and beta-carotenes, are always present in the leaf throughout the year during the summer. Just can't see them because chlorophylls, which reflect green light, are the dominant pigments in most of the deciduous trees or plants that lose their leaves each year. Most of those that you're going to see around Ohio have green leaves, and they have green leaves because the dominant pigments in terms of just amount of that pigment in the leaf is chlorophyll, and so it appears green. When temperatures fall and the chlorophylls tend to denature and degrade, the other pigments show up, the purples and the oranges and the yellows that are represented in carotenes, phycocyanins, phycoerythrins. They are all there, and you get to see them after the chlorophyll goes away. There's also an interaction between a carbohydrate production that is specific to the fall and the end pigments to come up with colors that you don't see all year, and those are the deep reds. And those are called, let me see if I can remember. Yeah, here we are. The anthocyanins. And that's what that link will take you to, is to this graphic. So these colors are showing up because this color is dying off, but this color is an interaction between existing pigments in the plant and sugars that are specifically made in the fall. The other question, why do trees lose leaves each fall? Of course, you have non-deciduous trees like evergreens that don't fully lose their leaves, their needles, although they may lose a portion of them in the fall. The deciduous trees, the quote-unquote reason that they lose their leaves is it gives them an advantage in terms of water loss over the course of the over the course of the winter. So in the summer, when there's a lot of sunlight to photosynthesize, it makes sense to express a leaf, which allows you to capture that light, and the amount of carbohydrate that you can fix compared to the amount of water that you will lose is a good balance for the growing season during the summer. But in the winter, you have less access to water because it may be frozen and so can't get to your roots. And you don't want to have all these pores in the stomata of the leaves, which will make you prone to losing that water. So instead, you drop your leaves and prevent that water loss, and you use the food that you made throughout the year to make it through the winter until the sun comes back out. There's also a risk of storms in the wintertime that such as sleet, freezing rain, which can add weight to limbs if it attaches to leaves and then cause plants to lose big limbs. So these are the cited reasons as to why plants lose their leaves. All right. So at the photosystem, which is on this diagram right here, you're going to pass off your electrons from the antenna chlorophyll to a reaction center chlorophyll. And there are two photosystems that we're going to talk about for most higher plants. Not all plants or not all photosynthesizers use this system, but this is the one we're going to talk about. They are named in the order in which they were discovered. So even though we're going to talk about this diagram from left to right, because it's just easier to consider that way, it goes photosystem 2 and then photosystem 1. This is the first one discovered. This is the second one discovered. P680 refers to the maximum action spectrum for that photosystem, and that is most activated at 680 nanometers wavelength of light, whereas this one has its peak at 700 nanometers wavelength of light. Okay, so if we look at the energy of the electron, so the more electronegative is on toward the top of the graph, less electronegative is on the bottom of the graph. These are high energy electrons and low energy electrons. Okay, we saw similar sorts of diagrams when we talked about glycolysis. The photon is going to come in. It's going to be have its energy absorbed by an antenna chlorophyll, which is going to use resonance transfer to pass that energy to another chlorophyll, and then eventually to the P680 reaction center, which will cause its electron to physically leave the chlorophyll molecule and get passed on to an electron carrier. And this is an electron transport chain very similar to what you saw in mitochondria, in stage 4 of mitochondria, where you're passing off electrons, and you're going to see the same result as a result of passing off those electrons, in that you're going to use the energy that these electrons are losing to move protons. Okay, so you pass off to these different electron carriers. Okay, some are embedded in the membrane, some are soluble, like plasticinin. And as these energies lose electrons, those energy loss is used to move protons into the lumen of the thylakoid. So the thylakoid outer surface is made up of membrane, just like the skin of your pancake. And then what's going to happen is protons are going to get pumped into the middle of that pancake, like you're having a stuffed pancake, stuffed with jelly or something like that, and you're stuffing it with protons. Okay, so they're moving from the stroma into the lumen. Then the same thing while this is happening, these guys are losing electrons. Same thing is happening in photosystem 1, that at the P700 reaction center, they're also going to lose electrons in the same manner, in that the antenna chlorophylls absorb photons. Those photons use resonant transfer to transfer the energy to other antenna chlorophylls until it gets to the P700 reaction center chlorophyll, and then that'll kick out an electron, physically removing it from the center. The electrons that leave P700 to go to this electron transport chain get replaced by the electrons coming from the ones from P680. Okay, so these electrons leave and they get replaced by these guys. Now we're going to go back and finish it up after we finish the electron transport chain here. But these have electrons that leave. That leaves a hole in P700 that gets replaced by electrons coming from P680. This passes down on another electron transport chain. These electrons lose energy at each step, and then eventually these are passed on to NADP to make NADPH. So don't get NAD and NADP confused. They are different animals. So NADP is the terminal electron acceptor for photosynthetic light reactions. Oxygen is the terminal electron acceptor for electron transport in mitochondria. Okay, so you pass off your electrons finally to NADP and you make NADPH. Okay, so let's fill in some gaps here. So these P680 electrons left, that leaves a hole in this molecule, and you can't keep doing that forever. You can't just keep ripping electrons off of P680 and have that continue. So you have to replace those electrons. Where do you replace those electrons from? You replace them from water. So you split water using a catalyst. You rip the electrons off of that water and use those electrons to replace the electrons that leave P680. The byproduct of that is oxygen. So you split the water into hydrogen and oxygen, use the electrons from hydrogen to fill the hole that's left in P680, and what's left over is oxygen. So oxygen is a byproduct of photosynthesis. It's a waste product. And for early life evolution on the planet, oxygen was toxic to a lot of the organisms that were living without oxygen at that time. And eventually there were mutations and adaptations to take advantage of oxygen. But high concentrations of oxygen are still toxic, even toxic to you. Too high a concentration of oxygen is toxic because it's a waste product. Okay, so I'm going to go over it in a different diagram instead of staying on this diagram. Once again, the last place that the electrons go in the light reactions is to NADP to make NADPH. That makes the terminal electron acceptor in the light reactions NADP, whereas the terminal electron acceptor in mitochondria in oxidative phosphorylation is oxygen. Okay, that should be easy. Okay, so one more time looking at it in a different diagram. So instead of an electron negativity graph, we have the light reactions on the thylakoid membrane. Here's the lumen inside of the pancake. Here's the stroma outside of the pancake. This is where the maple syrup is. And inside of the pancake, if you're having stuffed pancakes, this is where the jelly is. Okay. So electrons are absorbed by the photosystem. The photosystem 2 is the first one we're going to talk about. There are antenna chlorophylls in this photosystem and a reaction center chlorophyll. The antenna chlorophylls pass off their energy absorbed from the photons to the reaction center chlorophyll by resonance transfer. That causes an electron from the reaction center chlorophyll to be passed off to an electron carrier. That electron is replaced from splitting water. So water is split using a manganese catalyst. So magnesium, Mg, is the metal that is inside chlorophyll, but manganese, Mn, is the part of the catalyst that allows plants to split water. So the water is split, take the electrons from that water, replace them for the ones that left P680. Oxygen now is a byproduct. These high energy electrons are going to lose energy by passing from one electron carrier to another. Okay? And that energy loss is used to move protons from the stroma, where the maple syrup is, into the lumen, inside the stuffing of the pancake. Okay? Pass off to other electron carriers. Some electron carriers are membrane bound, some are soluble in water, just like you saw in mitochondria. These are passing off electrons to photosystem 1. Now, while this has been happening, photosystem 1 is doing the same thing as photosystem 2. They're absorbing photons, they're getting excited. Those electrons are leaving. The hole in photosystem 1 gets replaced by the electrons that came from photosystem 2. Okay? So the hole in photosystem 2 gets plugged by electrons from water. The hole in photosystem 1 gets plugged by electrons that came from photosystem 2. Again, these high energy electrons get passed along an electron transport chain. Eventually, they're going to get passed on to NADP to make NADPH. Then the protons that got pumped from the stroma to the lumen create a gradient of protons higher in the lumen, lower in the stroma, and those protons are going to go through an FOF1 ATPase. It's called CFO-CF1, but it looks and acts and operates just like the one in mitochondria. It's called C for chloroplast. Those protons are going to go through the FO channel into the F1 head. Those beta subunits are going to change shape 120 degrees per turn, and they're going to put together ADP and P to make ATP, just like you saw in mitochondria. But unlike in mitochondria that got their energy, those high energy electrons, from breaking down food, glucose, plants are getting their high energy electrons from the sun by just taking apart water and passing on electrons ultimately to NADPH, and then using the change in electron energy to move protons, and then using those protons to make ATP. It's pretty amazing what these guys can do without eating. You're going to see that these light reactions, these light-dependent reactions, because they depend on light, the products that they make, NADPH and ATP, are going to be used in a synthesis reaction called the Calvin cycle, also called the dark reactions, and that we'll talk about in the next podcast. So even though these are often called the light reactions, and that's what I'm calling them in this podcast, as soon as the lights go off, all of this stops. Okay? the whole thing stops. So this depends upon light for getting the electrons excited, but both light and dark reactions stop as soon as the lights go off. Again, just to remind you of the anatomy of the chloroplast and where things are happening. All of those light-dependent reactions are happening on this membrane. When it's stacked up, it's called grana, but there are photosystems all over this massively folded innermost membrane of chloroplast. Two outer membranes, and then this is the innermost membrane. And the space outside of those membranes is called the stroma. So during light-dependent electron transport, protons are pumped into the lumen, and then they're going to come back out into the stroma to make ATP. So protons are pumped into the lumen as a result of electrons moving on this membrane, and then to make ATP, they go back out into the stroma. Okay, so hopefully that'll help you answer this one. Now, this is one of my favorite stories, and if you ever take a tropical class with me, and I hope you get a chance to do that, this is a story that I tell over and over until people get sick of it, because you get to see these plants in the tropics. So plants also make hemoglobin, just like the hemoglobin that is in your blood. It's not exactly like it. Like we can tell the difference between plant and animal hemoglobin, but it's pretty similar, and it does bind to oxygen. And so why would plants need hemoglobin? They don't have a body that depends on the delivery of oxygen. Well, sort of, but not in the same way. The reason is in places where there's too much light intensity, like in the tropics, what can happen is the reaction center chlorophyll can react with oxygen. So oxygen is around because it's made as a byproduct. And photosynthesis is chugging along at such a rate that that reaction center chlorophyll can make a free radical. And free radicals, you may have heard about them before. These are essentially molecules with an unpaired electron, and they will take that unpaired electron and grab an electron from anything that's next to it, including things you might not want, like your DNA, and mutate your DNA. And so these pigments will suck up that oxygen, any excess oxygen, to lessen this process, and they tend to be at the edge of leaves here, where you see lots of potential for photosynthesis, because it's like the front line of where they're going to absorb light. And they can also accept excess electrons and dissipate those electrons as heat. So not the hemoglobin, but xanthophylls and carotenoids, other light-absorbing pigments. So it's not that the xanthophylls and carotenoids are sucking up the oxygen. It's the hemoglobin that is sucking up the oxygen. Oh, and another way that these hemoglobins have become famous, these plant hemoglobins have become famous, is in the food industry just over the last couple of years for these impossible burgers or these alternative plant-based meats, quote unquote. And these are using hemoglobins it's usually from peas, to give the appearance of meat as if it is bleeding, but that's just hemoglobin from plants. And also to give a taste that people expect Part of what that taste is when you're tasting meat is hemoglobin and also myoglobin, which is expressed in skeletal muscle. And when you're eating meat, you're eating skeletal muscle. So to reproduce that effect, you take hemoglobin from plants. Again, it's usually, if you look on the ingredients, usually they say pea protein, and what they mean is the like hemoglobin from peas to give it the appearance and taste and feel and even behavior of meat, but it's not meat.